Two forms of propagation exist. The first, continuous conduction, occurs by depolarizing the adjacent region of the axon membrane. The second, saltatory conduction, occurs by jumping from node to node. Differing the two is the presence of myelin or fatty material during saltatory conduction and the lack of myelin during continuous conduction. This myelin is formed by neuroglia, a non-neural cell. The latter, saltatory conduction, results in a much higher conduction velocity, or a faster speed of conduction, and thus will be the focus in this presentation. The whole point of propagation is to get the message from a neuron, the cell that transmits information, all the way down the axon or along cytoplasmic tube. No matter the length constant or the distance of the axon, the message will relay to the very end. All propagation occurs in the nodes of Ranvier. And these are the nodes right here along the axon. That is the region of exposed plasma membrane between segments of myelin. Sodium is on the outside and potassium is on the inside because of the sodium-potassium pumps. These pink and purple, which allows each to come through. At resting potential, both voltage-gated sodium and potassium ion channels are closed, and inactivation gates are open. These gates allow the entrances and exits of sodium and potassium. They are activated and inactivated in response to changes in the transmembrane potential. After depolarization begins, this makes membrane potential more positive, causing voltage-gated channels to start opening. They open as sodium moves across the membrane but at a slower rate. Depolarization happens because more sodium goes in a cell than potassium goes out. The sodium channels then open and sodium rushes into the cytoplasm. This is an action potential, a propagated, short-lasting change in the transmembrane potential. In response, the inner membrane changes from negative to positive. The movement of these positive charges is called a local current. The sodium channels are then inactivated and the potassium channels are activated. This lasts a while to bring the membrane potential back to resting point. Lastly, both gates are closed, re-establishing resting potential. The membrane resistances, also known as the leakiness, decreases. This makes everything pass quicker and easier. The oligodendrocytes, or neurological cells, are located in the central nervous system where the brain and spinal cord are found. The Schwann cells are found in the peripheral nervous system, which lies outside the brain and spinal cord. Both the oligodendrocytes and the Schwann cells then wrap an axon in myelin.